Hello? How are you? Hi.
Yeah, good.
Good.
How are you?
All right. So I guess if you wanted to give any updates, now's the time.
Sir, I just wanted to let you know that I've been reading and trying to understand whether what can be extended from like previous project, because I have seen that in the last meeting I've discussed that linear hypergraphs and lineage hypergraphs and the spatial hypergraphs. So my theme was to make a unified model, so which can integrate both, and we can design the dynamic like embryogenesis can be made in a in a dynamic fashion by both integrating both the features. So what I was thinking is I'll share my screen once. So it might be a little vague, but I am trying to think that I've been reading this work for a quite a long time. So this was a recent work, and I read up like certain parts of it, and I wanted to implement, because this works fundamentally focuses on various hypergraph neural networks. So how is the message passing done? How do we design the features? How do we design the structural information? How can we feed into the network? How do we define hypergraphs? What kind of different variations that we can see in this, and how can we do the message passing? How can we learn the temporal relationships for growing hypergraphs? So I thought this could be a great work that to start with. So what I'm thinking is right now, so first I want to design a unified hypergraph, a network which can both understand the lineage and the 3D spatial information. With that, I will be able to understand, so how does a hypergraph structure help us? And later on, I wanted to understand and apply different mechanisms such as the message passing mechanisms, and understand how attention and where exactly the attention is required, and how do the message passing evolve. Do we require what is the significant component while during the understanding the first connectomes, and then during the evolution, what are the key significant factors it's actually essential, and where it's actually the model is trying to depend. This progression I wanted to understand using these type of hypergraph models. So these are not yet fully benchmarked. So I wanted to read this work, and I wanted to present all the detailed information regarding the various hypergraphs, and I want to implement for the C. elegans evolutionary data. So I think, doing that, if I try it, I'm pretty not sure I'll be able to visualize well. But what I can do is, if suppose I wanted to attend at a certain, these are certain features and these are certain nodes that I'm trying to attend. Providing the attention, then I will try to visualize using some of basic tools. But my idea is to incorporate hypergraph neural networks into to see how the development progresses. I wanted to see the progression using these different set of methods, and I thought this could be apt into the space, because what we thought was to design of more like a diverse hypergraph architectures where we can implement them step by step, and first we'll come up with a unified model, and on top of that we'll try to understand various attention mechanisms that can be possible and another. And I also thought of why this could exactly align into the spaces, because what we were thinking is it's not that cell is they divide and they integrate, and during this process I think that the hypergraph, this multicellular interactions that we see, this and the group level transitions that may happen during the development, that can be definitely seen by understanding various mechanisms of this sort. So I try to get a clear intuition of this after reading, and I thought this could be a correct fit into the space. So I'll share the paper, sir, and probably I'll give a presentation. And while giving the presentation, I will maintain the perspective that how this could be useful for our the C elegans, how this can be useful for and each and every component that I'm trying to explain. I will probably explain in the perspective of how it can be useful for only to C elegans, the developmental process of embryogenesis. So
yeah, it sounds good. You weren't going to present it today, were you? Or?
No, sir. I am preparing the presentation, and probably by the next week I'll be able to finish the entire presentation.
Okay, that sounds good. Thank you.
Yes,
yeah,
sir.
It looks interesting. So could you bring back that? Could you reshare your screen? I wanted to see that.
Yeah, sure, sir.
Taxonomy, I guess.
Yes. Yes, Amen.
All right. Yeah, it looks interesting. So you have you're modeling higher order interactions.
Yes.
And you're encoding input features.
Yes.
Input structure, message passing,
and the way we are training the network is objective. Yeah. So there are two different mechanisms which I'm really interested in. One is a supervised approach where we give, saying that, this is the label or this is the ground truth, and we are trying to train in that perspective, or we can use a self-supervised perspective. So I've read the paper, but I wanted to delve into deep with a nice perspective of C elegans, and I think implementing in this fashion could be a next extension to the work that was already done, and I think this could be a great start off is what I felt. So I want your thought on this.
Yeah. Well, yeah, I think we go through the taxonomy and see which things are kind of fit the data the best.
Okay.
I don't know how you had this in mind. I would maybe propose the best things that you can think of from your perspective, and then we can argue about it or evaluate it. I think you should be able to break things down now. So we can make it. I'm curious, is this something that you are kind of aiming for in your proposal? I know you're writing a Google Summer of Code proposal.
Yes, sir. I have started a small description of it, but I'm curating the references, and I'm thinking that what actually aligns and what actually makes a good improvement. So I also wanted to see it in not just the code perspective, but I wanted to see that what actually this can get me. So I also want the novelty part to be done. So that will give me more motivated, keep me more motivated, and work through the lines. So if it's going to be a good publishable results, then that will be very much helpful for me or in even the community.
Okay. Yeah, I think. We'd have to talk about that further.
Yes.
So the supervised versus unsupervised perspective. You're talking about the kinds of data that you can bring into the algorithm. Yeah. So C. elegans has labels and a lot of. I think you could have a lot of supervision there potentially.
Yes.
You could have labels of cells, or labels of tissues, or labels of time. We have all sorts of data like that. There are other systems where you don't have that, and that's why we work in C. elegans a lot. But
yes,
there are other ways that you can do this that aren't labeled necessarily, or the labels change, and it's harder to get good label data. So it really depends. If you want something that's just specific to C. elegans, supervised is good, but sort of biologically generalizable, I guess you could say, unsupervised would be good. So
Yes.
things you have to think about like that. And
Yes.
yeah, I think this is all good.
Okay.
We'll go over that next week, I guess.
Yes, and I'll start writing, a rough draft probably, so that we can iterate on top of that.
Rough draft of what?
A rough draft of the proposal of how do we take things. I mean, like what could be a good weightage to the project,
Yeah. Okay.
Okay.
All right. So, do we have anyone else wanted to give an update? Thank you, Lalith. That was good.
Thank you.
Jayadratha or Soham.
Yeah, I want to ask one thing. So the mail you have sent that has all the data sets that are available, and I checked all those data sets and I tried to understand. And those are already available in GitHub, DevoWorm, GitHub page, right? Most of them, the data sets. Yeah. So I analyzed those data sets, and the last meeting, so we discussed about the temporal graph methods now, and also we have discussed about visualization thing in 3D. So yeah. So the thing is, I understood about from the data which are already available from that, this temporal graph part that can be done like as I can understand, and for the visualization part that can be a side project or like small thing that we can include, but for a larger project or for this GSA, I understood like this can be better to pursue the temporal graph part. So what do you think? I want your opinion on that. Should we include both of them, like the visualization, based on our discussion last day? Is it the visualization as well as the temporal graph part, or only temporal graph part for this GSA better?
So I think that you could have both. I think both, but I think the temporal graph stuff would have to be kind of the priority. Then the visualization could come kind of at the end. Like, so, getting down the basics of what that's going to look like, implementing it, and then visualizing things. Because it's hard to visualize something you don't have the methods nailed down for. We have, I don't know how much work the visualization would take. Maybe a couple of weeks to get something. I mean, you could work on it as you're going along, but I would prioritize the temporal graph part, and then maybe try some visualizations at the end, or try to implement some. Usually, the way we do proposals is we say, what do we want to focus on, and then what do we want to sort of follow up on later. And then, if we have time, we can implement the visualizations fully. But just having like kind of a, sort of this is what it would look like. It's just kind of you're not spending all of your time on the visualization. Does that make sense?
Yeah, understood. Yeah, thank you. Thanks.
All right. Yeah, thank you, Jayadratha.
Hi, Bradley. Sorry to interrupt. I don't really have an update. Just wanted to say I have written my proposal draft based on the presentation I gave last week. So I will send that to you via Slack. So if you have time, please give your feedbacks before submitting the final.
Okay. All right. Susan, how are you?
Hello. All I've got is interesting vacation photos. So, yeah.
That's good.
Anyways, I'm just recovering from a vacation, so
Excellent.
I have to write a paper, so I need to get on with it. Yeah.
All right. Thanks.
Okay.
Oh, Mehul, how are you?
Hi Bradley. Hi everyone. Yeah, I'm good. This week, like I was talking to like Lalith, Soham, John, and Soham has like given like a rough draft. Like I still have to review it. Like he gave me like I think yesterday, so I'll get back to him. So that is what I'm up to these days, like on the weekends.
Yeah. All right. Sounds good.
Yeah.
Anything else?
Yeah. No, Bradley, nothing else right now.
All right. Thanks.
Thanks. Yeah.
Hello, Morgan. Hello, Dick. All right. So let's start off with some things. So I have another article on this sort of theme that we've been talking about a little bit, called "Limits on a Cell." So we did this. We covered this Asimov press article on breaking the speed limit of cell division. That I don't remember when that was. I think it was in January. And I went through this paper, or this article, talking about sort of the limits to cell division. So the idea was that, if we want to have a programmable system, we can think about a computer program and how fast we can run computer programs. And then we might want to take that sort of framework and apply it to biological systems. So like for things like synthetic biology, where you want to program cells and have them reproduce, produce like, a population of things you can use for different purposes, like you might do with programs as well. How do we do that? And what are the limits? And so in this paper, they talk about the biophysical limits of that kind of an approach. You can have, say, like a population of bacterial cells. You can, boot them up with different genes, and then you can use them for different things. But to get, like, say, a certain number of cells, or in this case, I guess they're making the analogy to programs, you have some biophysical limits. And some of those limits are DNA replication, and then more generally the speed of cell division. So the speed of cell division involves replication of DNA, replication of the genome, and then the process of the cell dividing and everything kind of forming in the new cells. So that's what they're talking about in this article. We went through this, where they talk about the biophysical limits. They focused on the synthesis of ribosomes, because those are the things that you need, sort of the key components you need to make proteins. So you have to duplicate your genome, you have to create ribosomes, you have to synthesize those. Then you have to have ribosomes, enough ribosomes in each cell to make proteins. And then you need to have the membrane sort of enclosed in both cells. And so all that takes time, and there's a fundamental speed limit for DNA replication and for ribosome synthesis. And so that's the limit that they have. And so if you're thinking in terms of how to make this process as fast as possible, then you're limited by this biophysical barrier. So you can't necessarily increase the phases of cell division, for example. So we have, a G1 phase and G2 phase, and we have so they have these intermediate steps between rounds of replication and division, stalling division by roughly 15 more hours. So you have to go through G1 and G2 phase to get to cell division, and that's 15 hours, where you can't shorten that time period. And so, sometimes you have cell types like HeLa cells, which actually are cancerous cells that can double much more quickly than a typical cell. But even there, you're limited to a speed limit. You can't do this instantaneously. And then we have bacteria that are very slow. So depending on the bacterial cell you choose, it can be even slower than that. But there is this fundamental speed limit for cell division, and it has to do with cell cycle, replicating the genome, and synthesizing ribosomes. Now that was about the speed of cell division. So that's how fast can you make cell division, and it varies in different species. It varies in different types of cells, bacterial cells versus eukaryotic cells. Bacterial cells are faster because they have to produce fewer subunits for their ribosomes, and their genomes are generally much smaller. And so eukaryotic cells are even slower, or even have a higher or a lower fundamental speed limit. Now this article is another article from Asimov Press, and now we'll talk about cell size. So this is what limits a cell's size, and this is where we talk about two physical constraints help explain why cells are so tiny. The first is the surface area to volume ratio, and the second is diffusion. So I think this is something that maybe we're more comfortable with in this group. We've thought about this more, I think, than the other example. But it's, they're both pretty important. So before we thought about, okay, how long does it take for a cell to divide? What's the sort of the fundamental doubling time? What's the fastest doubling time you can have in a cell? And, we can do those calculations. And of course, you can't speed that up because you need to have those components go through those steps that we talked about. In terms of a cell size, we have these physical constraints. It kind of act in the same way that our constraints on time act. So surface area to volume ratio is where you have a surface area, which is the membrane, and then the volume, which is the inside of the cell. So as cells grow in size, their surface area grows, and so we know that cells in general are pretty small, but sometimes cells can be very big, and this is something we've talked about with respect to the embryo. How big can you make a cell? And of course, if you think about why don't you just have one big cell doing all sorts of things within the cell, I think the answer to that is actually pretty easy. Is that we want to have differentiation in different parts of the embryo, so it needs to be multicellular. But at the same time, we could have a single cell organism that emerges. We could have very large single cell organisms instead of multicellular organisms. And so, why not? So one constraint is that surface area to volume ratio has to remain small or reasonable. And then diffusion, which means that if you have something that's generated like a bunch of molecules that you want to reach every part of the cell evenly, then it can't, if it diffuses by something like Brownian motion, then it can't go very far over a certain amount of time. So there's a speed limit as to how things can diffuse. So if you have a really big cell, it takes a while for things to diffuse and get out there. So we'll talk actually about, I have a paper that just came out this week on Archaebacteria and how there's actually, under some conditions, you have multicellularity. So we'll talk about that. But also this, you might also think about well, why don't we have really big cells? And of course, in Drosophila, we have in development this syncytial state where you have this big cell with a bunch of nuclei. And then at some point, you'll have the cellularization phase where things break down and things become smaller cells. And each of those nuclei become, have a host that's, or a cell hosts each nucleus. So, and then we talked about syncytium or maybe 2 weeks ago, where, those are where you have multiple nuclei in a single cell body. But again, we talked about what that would look like. It isn't necessarily this big sphere. It's kind of this thing where the cell bodies are connected, and we don't know exactly. There are all different geometries that you can have, so we don't know if that huge cell would be feasible. And then we can have things like protocells, which people use in early life research, where they just have like this big vesicle that they put things in, like RNA and proteins, and they just use that as a bag of replication. And I don't know too much about if they've experimented with the size of those vesicles, but they're generally not too big. But anyways, so this is what limits a cell size. So this is this article starts with a human body is built for 30 trillion cells, excluding microbes, that each arise from a lone fertilized egg. These cells come in a multiplicity of shapes and sizes, with internal volumes spanning 5 orders of magnitude. So this is where you have cells that, depending on the cell, if it's a microbial cell, if it's a mammalian cell, if it's a amphibian cell, they're all different sizes, and that spans 5 orders of magnitude. So the smallest human cell, a sperm, fills a volume of just 30 microns cubed, whereas an oocyte boasts a volume of 4 million microns cubed, making it the largest cell in the human body. So an oocyte is actually very large compared to a sperm, and different somatic cells have different sizes, and there's a wide range there. So then what accounts for this huge range, even within an organism? And of course, we know, again, that like this varies across species, but also within species within the same organism sometimes. A simplistic answer would be that evolution has simply made each cell whatever size best suits its function, which would make sense if you're thinking about something like sperm. It's a specialized cell. It's small because the body produces many of them, and tinier because it costs less energy to make and maintain. Sperm are also ultra minimal, stripped down to little more than DNA, and the few mitochondria necessary for providing energy to spin their whip-like tails. So they don't do a lot. Functionally, they're very specialized, and so you don't need a lot of stuff on board. By contrast, an oocyte needs massive reserves of mitochondria and nutrients to support early embryonic growth. In short, every cell is as large or small as it needs to be within reason. And then, this is where basically you have these physical constraints. One major limitation on a cell's size is the surface area to volume ratio. So assuming a cell is roughly spherical in shape, its internal volume grows proportionally to the cube of the sphere's radius. So this is why we're using these micron cube measurements, because we have this cubic measurement that we want to understand. So the internal volume within the cell, the sphere, it grows to the cube of a sphere's radius. And so we're looking at the radius and kind of, everything kind of, we assume a symmetrical cell first of all. So it's moving outward as, I guess we need space in the cell. And its surface area grows proportionally to the square of that radius. So we're thinking about the volume, and then we're thinking about the surface area, which is the membrane. And so the membrane, of course, is important because things happen at the membrane. Proteins dock at the membrane. You get signaling molecules that dock at the membrane. All sorts of things happen there that are this interface between the inside of the cell and the outside of the cell. So having that surface may or may not be very important for different functions. So, in other words, the cell's volume grows much more swiftly than its surface area. So, just by sheer geometry. This ratio has serious consequences for cellular survival. The cell's membrane funnels nutrients into the cell and secretes waste. It's also where the energy in a prokaryotic cell, like E. coli, get made. So this is where, in E. coli you have things happening at the membrane, like energy production. You also have waste removal and nutrient inputs, and then, of course, signaling and other things. If the interior grows too large relative to the membrane, the cell's metabolic processes slowly will crawl. So this is where, if we have this really big cell, huge cell, and then the membrane is, minimized, then that slows down metabolic processes because there's no space for them to proceed. I guess that's what's getting in here. And then this shows a graph. A cell's volume scales faster than surface area. So the internal volume of a spherical cell is proportional to the cube of its radius, whereas its surface area is proportional to the square of its radius. And this is for the graph. So for very, very big volumes, you would have a reduced surface area relative to that. That's what he's getting at with the ratio. And so, the larger the volume, the relatively smaller the surface area. And so to support all that volume, to support the metabolism for all that space, you don't have the surface area for that. And so metabolism falls until you get to a point where it's not viable. So that's why probably we don't have really big cells. Although in some cases they're specialized for certain functions, we might have much larger cells. But there is this limit to what we can have. Okay, so a second constraint is diffusion, or the tendency for molecules to migrate from areas of high concentration to areas of low concentration. If you know about basic diffusion from physics, you know that there's like a source, and like molecules will diffuse from that source, and they'll diffuse according to some function, and that function will describe sort of the path that these molecules take. They could take like a Brownian or random walk and do Brownian motion away from the source, but that also means that the closer you are to the source, the higher the concentration of the molecule. So you end up with this decay over space, which kind of decays linearly from the source usually, and then you also have this decay over time, which is where over time you get more molecules. They diffuse outward. Your area of diffusion grows, but then you have this decay process that can also happen at the edges, where these molecules, like maybe they have a half-life of whatever amount of time. So once you reach the half-life, they decay and they no longer become viable. So this source keeps pumping out these molecules. They diffuse outward, and of course they're limited by half-life, but they're also limited by sort of the density that we have. And so that's what diffusion basically is, and that's the constraint. We have this migration of molecules, but we also have this process of diffusion. So in terms of migration dictates how quickly enzymes find substrates, how effectively signaling molecules reach receptors, and how often ribosomes collide with messenger RNAs. So when we think about diffusion, we have to think about maybe where something is produced at a certain point, at an origin, and then diffuses throughout this volume, this internal volume. And then, of course, this migration doesn't just happen because it happens because there are things out there that might bind to this molecule. So I don't mean to say that there's like a purpose to this, but over time, you would have this sort of situation where there's something that's diffused, and if it's going to be functional, it's going to bind with things in the cell volume. And if the cell's too big, you might have sources of diffusion in different places. It's pumping out all these molecules, and it's not binding to its receptors, and it's not making this sort of intentional, I guess we could call it a deterministic walk, where it's guided towards its binding target and finds the quickest path. There's this random walk where things diffuse, they just diffuse outward, and then eventually they reach their target. So what that means is that you have to have a relatively constrained volume for that system to work, for things to be both in the state of diffusion via random walk and being able to bind to their targets, reach receptors, and collide with messenger RNAs. Now, granted, the inside of the cell is very packed, so it doesn't take a lot of intentional effort to do this, or it doesn't take some complex process. It just requires some Brownian motion. But that's why if things are very big, you can't have things packed that tightly so that it enables that sort of function. And in a smaller cell, things are packed in more tightly, so you can have these opportunities. Inside a cell, nearly everything happens by chance encounters amongst these molecules. So chance encounters, I think, is the key word here, or the key phrase. As the cell volume grows, the chance for any given encounter diminishes, assuming the numbers of molecules stay constant. And so the diffusion rate of molecules hinges on several factors, and this is also the other important part, is that molecular diffusion is not a constant, it's Brownian motion, but the rate at which it's produced and diffuses is also, variable and can play a role. So you can have larger cells with a higher diffusion rate, and the diffusion rate can keep up to a point. So for instance, the cytoplasm is extremely crowded, so molecules spend lots of time ricocheting off obstacles, delaying their arrival at a distant location. Every protein in a cell collides with about 10 billion water molecules per second, on average. These frequent collisions mean that the vast majority of proteins in a bacterium, for example, only diffuse between 5 and 10 nanometers squared per second. So that's, with respect to the size of the cell, it isn't that large. But again, if there are a lot of things packed inside the cell, then that's enough space. Some molecules also aggregate or stick to the charged surfaces, further slowing their movement. Molecules move very slowly near the densely packed nucleoid, for example. In general, larger molecules diffuse more sluggishly than smaller ones. So it gives the example of metabolites in E. coli, which can diffuse from one side of the cell to the other in milliseconds, which means collisions and cellular outcomes occur quickly. So, there are these, in C. elegans, you have this mechanism where by the rotor, the flagellar rotor moves, and you have these molecules that go from the sort of the head to the tail, and they kind of bounce around the inside of the cell. They have to bind with the receptors on the inside of the flagellar motor to make it work. And so this is something that happens through basically by chance, through diffusion. And so that's an example of some function that emerges from this sort of diffusive process. Metabolites are another example where you get this sort of, these collisions that have to occur, and, they can occur by chance enough to make something happen in the cell. A typical protein takes just 0.01 seconds to traverse a bacterium's diameter, about 1 micrometer or micron, but the same protein would take around 4 minutes to move 1 millimeter and more than 6 hours to move 1 centimeter. This is in part why cells are so tiny. So this graph is measured diffusion rate versus molecular weight. So this is an E. coli, and this is where we have proteins of different diffusion rates and molecular weights. So each protein's diffusion weight or diffusion rate was measured in cytoplasm using fluorescence correlation spectroscopy. So these are the different proteins here labeled, or the dots are proteins, and we have some labeled proteins. And then we have this constant diffusion rate function, microns per second squared, and that goes downward as we go along this x-axis, which is diffusion rate, and then we have our mass, which is kilodaltons, on the y-axis. And we see that as the mass decreases, the diffusion rate increases. And as the diffusion rate decreases, the mass increases with some variation. So some proteins deviate from this function more than others. But basically the idea is that at the larger or the heavier the protein, the molecular weight, the slower the diffusion rate. That makes sense. If you have something that's lighter, it travels faster. If you have like ping pong balls versus like a ping pong table or a ping pong paddle, if you kind of threw them up in the air, you kind of let them kind of diffuse. If they got caught up like in a tornado or something, there'd be the diffusion rate might be higher. So this is a good example of how this works. And this is just a physical constraint. So this would happen in any organism cell, and this happens uniformly across cells. The diffusion rate of it depends on the medium, I think, as well. So there's some difference. But usually the medium inside the cell isn't like, I don't know if there are huge differences in diffusion rate across different cell types. But anyways. So what with these constraints in mind, we can begin to speculate as to why various cells are shaped the way they are. Red blood cells are tiny and shaped like bioconcave discs. This aids with diffusion, so it's the shape of the actual thing that's diffusing. By abandoning a spherical shape and evolving towards more of a donut, they increase their surface area without compromising their compact volume. This in turn enhances their ability to exchange oxygen with cells in the body. Their small size, just 8 micrometers across, also helps them move through the narrow capillaries. In contrast, oocytes can grow so large, around 100 micrometers in diameter, in part because they are less metabolically active than other types of human cells, and thus don't depend so much on random collisions. So they're actually less metabolically active by nature, and so they can have this very large size without losing much function. They stockpile nutrients during oogenesis to wait out fertilization. Eukaryotic cells also grow large in general because they evolved compartmentalization. So they modularize specific functions into organelles. And so this limits the amount of diffusion that needs to happen locally for some function to happen. And it also allows for larger cells. So that's good. And that's basically the article as I wanted to go over. So let's see, we have some things in the chat. So Dick says that there is at least one archaea that seems to have no control over cell size. That's interesting. Some archaea make transitions between spherical and flat polygonal. I've calculated for one cell the volume reduces by a factor of 68. It's also interesting. Blood cells become cup-shaped in capillaries, possibly increasing gas exchange. Yeah, that was what we're talking about in the example. Okay. Yeah, so that's good. Any other questions? Okay. Yeah, so I mean, it's an interesting article, and viewed in light of the other article in terms of the speed limit of cell division, I think that's really interesting. Asimov Press, they have this series on quantifying the cell, so I thought that was really interesting as to how they're kind of approaching this. So actually, I wanted to point out a paper that Susan sent, and I don't know if we covered this before, but this is a paper from PNAS. This is on cell contraction induces long-range stress stiffening in the extracellular matrix. So this is, I don't want to cover this week, but we can cover it at another time. This talks about how animal cells and tissues are supported by biopolymer matrices, which typically exhibit highly nonlinear mechanical properties. We don't know how cells in turn affect nonlinear mechanics of their surrounding matrix. So in this paper, they show that living contractile cells are able to generate a massive stiffness gradient in 3 distinct 3D extracellular matrix model systems. So these collagen, fibrin, and matrigel, which is artificial type matrix. We decipher this remarkable behavior by introducing nonlinear stress interference microscopy, which is NSIM, a technique used to infer stress fields in a 3D matrix for nonlinear microreology measurements with optical tweezers. This is a specific type of force microscopy they're using. Using NSIM in simulations, we reveal large, long-range, cell-generated stress capable of buckling filaments in the matrix. So we can have these cell-generated stresses that are long-range, large stresses that can buckle filaments in the matrix. These stresses give rise to the large spatial extent of the observed cell-induced matrix stiffness gradient, which can provide a mechanism for mechanical communication between cells. So this is, did you have any words on this, Susan, this paper?
Words. I don't know. I do know the matrix or the basement membrane has what is called a J curve. Like it changes from changes slope. The elasticity changes with increasing strain or stress. So it's starts off at one slope and then changes, so it's a steeper slope. It's a sort of a J shape on the stress strain curve. So that affects the elasticity of the overall tissue, which is important in development for sure.
Yeah, that's good. Yeah, so this is just kind of showing these. They have an experimental test bed. They show kind of the range of different forces that are transmitted, long-range cell-generated stresses. So just saying that, can we make changes to the filaments by kind of inducing stresses? And then, this is something that happens in the cell where they're contracting kind of like due to maybe some environmental force or some signal that they're getting from other cells, and then they're able to produce these large-scale forces.
And that's another way of cells communicating with each other, so that they can use it.
Well, that's good. Thank you, Susan. I mean to call on you, but
Oh well, that's okay. My thesis shows that it's the attachments cells have to the basement membrane and to each other are important for the overall tissue elasticity.
all right. So I don't know. We might go over that paper in more detail next week. I just wanted to bring it up now. So the last thing I wanted to talk about today is this paper on Archaebacteria and how we get these almost tissue-like structures. It's, I guess, a form of multicellularity. And multicellularity is interesting because you can achieve that in numerous ways. It's not a hard and fast thing. It's one of the transitions of evolution that we have, but it's also you can achieve it in multiple ways where we have this sort of tissue-like multicellular development. So it's not a tissue, but it's like a tissue. It behaves like a tissue. And this is triggered by mechanical compression in archaea. So this is where you have this mechanical compression that you can introduce, and it can form these structures that are, they're clonal, what they call clonal multicellularity. Okay. So this is in, I think it's in Science. Yeah, it's in latest issue of Science. And the abstract reads, the advent of clonal multicellularity is a critical evolutionary milestone, seen often in eukaryotes, rarely in bacteria, and only once in archaea. So this is one of these transitions of evolution where you go from like single cells living in isolation. Maybe they'll live in, there's a kind of a fuzzy boundary where they live in colonies, but the colonies are not, integrated in terms of, what I think we've talked about this before, in terms of metabolism and other things. And then eventually you get these structures that are multicellular, where they're, actually dependent on one another. And then, of course, you get embryos and things like that. So this is a major transition in the evolution of life because it allowed for these multicellular organisms to emerge. And so you don't see this, you see this sort of in, yeast are eukaryotes, and people have done a lot of work in yeast. Yeast are eukaryotes that are single cell, and they are single cell usually, but they form colonies, and moreover, they form these kind of clumps or clusters that are sort of multicellular, because you can induce multicellularity. In bacteria, you don't really see multicellularity as much. You see some, and then this is talking about archaea, which is, not bacteria, it's Archaebacteria, so it's very rare in Archaebacteria. In this case, what they're doing is they're inducing multicellularity. So they show that uniaxial compression induces clonal multicellularity in haloarchaea. This is just a specific type of Archaebacteria forming tissue-like structures. And then they say tissue-like, it's something in biology where you don't want to go all the way and say it's a tissue and have people try to confirm it's a tissue through experiments. Then you just say tissue-like, and people will know kind of what it's supposed to be like, but it's less rigorous. In any case, it's tissue-like, so don't get too excited. These archaeal tissues are mechanically and molecularly distinct from the unicellular lifestyle in making several eukaryotic features. So these have sort of these eukaryotic features that emerge once they're this type of multicellular structure. So it suggests that we have this sort of dependence that emerges, not due to any inherent feature of eukaryotic cells, but it's an inherent feature of multicellularity. Archaeal tissues undergo a multinucleate stage, followed by a tubulin-independent cellularization, orchestrated by an active membrane tension at a critical cell size. After cellularization, tissue junction elasticity becomes akin to that of animal tissues, giving rise to two cell types. They call peripheral cell types, central scutoid cell types, with distinct actin and protein glycosylation polarity patterns. So this means is that you have these multicellular collectives, tissue-like, and they form these different cell types. Like basically, the cell types are driven by this sort of these mechanical forces, this tissue junction elasticity, and different aspects of membrane proteins configuring themselves in different ways. So we have these peripheral cells and central scutoid cells. So it's like we have the central zone and a peripheral zone. So we're getting this sort of specialization already in these kind of structures. And there are different distinct patterns of glycosylation and polarity. Our findings highlight the potential convergent evolution of a biophysical mechanism in the emergence of multicellular systems across domains. So, the question I have here is this something that is unique to these cells? It's like a behavioral mode of this species of Archaebacteria, or is this something you can induce in all Archaebacteria? And I suspect it's maybe a function of the specific species that they have here. So let's see what we can find here. So they talk about multicellularity, and they talk about Archaebacteria as how they're distinct from bacteria, but they have this. So basically, archaea are now recognized as a monophyletic group with eukaryotes, so they actually are closer to eukaryotes than they are to bacteria. Most archaea lack a rigid cell wall and are encapsulated by a proteinaceous surface monolayer, S layer, a two-dimensional paracrystalline lattice composed of glycoproteins. So this is this S layer. And so they don't have a rigid cell wall, but they have this S layer. So that's the thing that's different. So, and I say that Archaebacteria are closer to eukaryotes and bacteria. They're just this monophyletic group. So they're going to be very different than eukaryotes. It's just that their classification is such that they're grouped with eukaryotes. So this might suggest that you see some properties of eukaryotic cells in Archaebacteria. But anyways, so you have, although the archaeal envelope structure is thought to make cells mechanically vulnerable, so this specialized, or this envelope structure, instead of having the rigid cell wall, it may make cells mechanically vulnerable. It also facilitates close interactions between cells, such as cell-cell contact and fusion, which may have played a role in the emergence of eukaryotes. So this is interesting because it suggests that this monophyletic origin sort of means that Archaebacteria, or the common ancestor between Archaebacteria and eukaryotes, also had something similar to this sort of S layer instead of a rigid cell wall, and then the rigid cell wall evolved independently in eukaryotes. So we don't know that, but that's kind of what it's suggesting. So this S layer may be, a sort of a stand-in for a rigid cell wall. While the archaeal envelope structure is thought to make cells mechanically vulnerable, we talked about that. However, the evolution of mechanosensory responses, driven by the lack of a rigid cell wall, remains elusive, owing to the scarcity of in vivo studies. A peculiar combination of genetic and biophysical traits prompted us to investigate the mechanobiology of archaeal cells, leading to the serendipitous discovery of a reversible clonal tissue-like multicellular developmental program. So, this is interesting. So there's this, they propose to be a developmental program, a multicellular developmental program in archaeal cells, or in this mechanobiology of archaeal cells. So they perform their experiments in the salt-loving Alopherax vulcani, which HVO, which is a specific species of Archaebacteria, leveraging its straightforward cultivation and genetics. So this is a model organism within Archaebacteria. We don't know, I guess, if this is true across all Archaebacteria, but in this species we do have this sort of program. First, we established a baseline for mechanically unperturbed helioarchaeal cells trapped within an arc cell, custom microfluidic device. It's where you put a cell in a device where you can introduce different types of laminar and turbulent flows and measure the effects of different liquids and chemical compounds. You can fix the cell and have these flows and do things like that. So you can actually get some good, interesting measurements of mechanical interactions. Cells growing in arc cells show cell morphologies comparable to those of bulk liquid cultures, indicating no mechanical stress. Next, we image cells under agarose pads, a standard technique in microbial immobilization. So they used arc cell, which is the microfluidic device, and they used agarose pads, which is sort of the control. These agarose pads deform cells within a single generation, which is about 2.5 hours, at the lowest agarose concentrations, making pad immobilization incompatible with prolonged imaging. To quantify the compressive forces deforming cells, we measured the pad elasticity. So they just talked about their method here. Storage moduli of 0.25 to 3.5% pads revealed resistance forces of around 10 kilopascals, and those are sufficient enough to deform HVO cells. These values suggest that HVO cells may have viscoelastic properties close to that of eukaryotic cells, such as amoeba or mammalian cells, but orders of magnitude lower than those of most cell-walled organisms. So cell-walled organisms are like plants and other types of eukaryotes that have rigids, like, we have plants and animal membranes. So basically plant-like membranes. Given the mechanical sensitivity of haloarchaeal cells under pads, we tested these responses of HVO to compressive forces above 100 kilopascals, closer to forces experienced in their natural habitat, such as the human gut or salt bones. Following compression under pads with agarose concentrations of 1.5% and higher, cells stop dividing but continue to grow. After around 12 hours, cellularization occurs via simultaneous septation events, resulting in epithelial-like monolayer structures. So this is where we've introduced these compressive forces above 100 kilopascals. These are forces that you see in nature, and then you have this compression. I guess this is where you observe this sort of cellularization after about 12 hours, and you end up with these monolayer structures. These tissue-like ensembles resemble radial tessellation patterns in leaf tissues and multicellular green algae. Again, this parallel with eukaryotic cells and algae. And so you have this sort of, you get different types of formation, sort of physical responses to the same thing. So this suggests that maybe there's some parallel, or that they're very similar sorts of structures that have some common evolutionary ancestry. The morphological development before cellularization also resembles the coencytic phase of chytrids and chicken embryogenesis, in which cells multiply their nuclei without cell division. So this is again this sort of syncytial developmental stage. So this is where you get this, this sort of another parallel with eukaryotes. Time-lapse imaging of cells expressing neo-green pCNA shows continuous replication during development, supporting a coencytic-like phase preceding cellularization. So then animal and plant cells often sense and activate biochemical pathways in response to surface curvatures, material properties of the membranes. To test influence or material properties of the substrate and surface curvature of the membranes. To test the influence of the pad stiffness, we immobilized HVO cells under and on top of the same agarose pad. As a result, tissues are observed exclusively under the pads, suggesting that stiffness alone is not sufficient to induce multicellularity. So this is where we don't just need stiffness, we need something else. Furthermore, more cells compressed by different biolayer cake pad setups. So they're compressing with these two agarose cakes, pushing them down. They're, stiffness isn't enough. So what else do we need? You develop into tissue-like structures only when under compression by at least one stiff surface, ruling out a specific role of the cover slip other than providing a rigid surface for compression. Finally, tissue formation is independent of gravity, pad mass, or thickness, consistently initiated at the same coencytic area regardless of pad density. So these analogous features suggest that the archaeal tissue development represents an evolved biological program response to compression. So again, it isn't enough to have stiffness, you need compression. And then they did some other experiments here. These are some images here where you get these. So this is where they have PDMS, which is the microfluidic environment, and then the agarose pads, which are the compressor. They trap them in the PDMS, and then they compress them with agarose pads. They allowed the cell to sit there, and then there was diffusion of the cell between the pads, and then they show these different morphologies of the cells. So here are the trap cells in the PDMS, where they're kind of coming together in these colonies. You have the compressed state by the agarose pads. So this is where we have the unicell, the coencyte, the cellularization, and the tissue. So you see there are these different stages of multicellularization, where you have cellularization, then you have this tissue formation. You have then this, this is another one where you have the S layer, and then that leads to the tissue stage there, where you have cellularization.
It looks like it might be crack formation.
Yeah, maybe. I don't know what's going on there, but yeah. And then they just show some other things from their experiments. They have membrane displacement in microns, time after ablation in seconds. So you can see they did some ablation experiments, and they had this membrane displacement for the tissue versus the unicell. The tissue, you get much more membrane displacement after ablation. So that's, I'm not really sure what they were doing there. I don't want to go over too much of, the detail of what they did, but they basically did laser ablation of tissue regions. And so that, I guess you take out like a piece of the culture, and then you have to fill in, I guess, because tissues will fill in. You can have cell division that fill in gaps in a cell culture, but if you have like a tissue layer, it actually fills in, and the membrane gets displaced in a different way. So this is maybe what's going on here. And so this just shows again the tissue versus the unicells. The tissue have the structure. We have the two different cell types, the edge cells and then the inner cells here. And then this is the unicell, where you don't have that sort of functional distinction. It's just a collection of cells in a cluster. So you can see the difference there. Okay, so that's figure 1. And so then they say archaeal tissues are widespread in helioarchaea and counter-correlate with biofilm production. So this is interesting that they want to know, like in helioarchaeal tissues, what is the evolutionary diversity? So they constructed a phylogenomic tree spanning 57 genera, representing all helioarchaeal orders, imaging compressed cells from 52 species across 14 genera. Regardless of cell size or growth rate, 61.6% of tested helioarchaeal species form tissues. That's a pretty high rate for, so this is something that you see across this helioarchaeal clade. So it's like this group within Archaebacteria, it's helioarchaeal or salt-loving, and then 61.6 of those species form tissues with at least one instance of tissue development or no multicellularity in every tested genus. So this is something that's both common but also commonly lost in different genera. So this is something that is, it's obviously maybe something that's shared by the common ancestor of Heliobacteria, or Helioarchaea, but it's not, maybe it's environmentally contingent. Among non-forming tissue strains, we identify cases of cell death, shape deformation, and unnoticeable shape deformations under pads, or the agar pads. We also observed 3 strains, HTG salina, NCC Jayadratha, and HK Jayadratha, that exhibited aggressive multicellularity, similar to that of Methanosarcina, the only previously reported multicellular archaea. These results suggest that archaeal tissues emerge early in helioarchaeal evolution and remain dominant in the sample diversity, as I was saying. Next, we focus on strains from the Halopharax genus, in which HFX pravalens produces large deformed tissue structures, whereas HFX mediterraneae and gibsoni failed to develop growing instead of stacked colonies. So you get these different types of morphologies, and this suggests that this sort of multicellularity maybe, isn't as tissue-like as we like to think, because you get these different maybe it's like an intermediate between colonies and tissues, where you get these different morphologies that are sort of maybe ad hoc tissues, or something that is both resembling a colony and a tissue. So when they fail to develop, they grow these stacked colonies, and that's another piece of diversity there that's interesting. Although all three taxa are closely related, HMET is placed on the long branch, which means that there's more evolutionary space between that and the other members of its genus, suggesting an extended time for adaptation following the loss or gain of genetic material. So this is, again, they do a lot of evolutionary studies here. This shows the phylogeny, where they've kind of, they know the phylogeny, they did the experiments in all these species within this genus, and they saw these results. And so this is, again, we get this S layer, you get this growth over time, and you get these different morphologies. Okay, and then figure 3 shows that cellularization is independent of FITZ-Z's and FITZ-ZS and results in 2 cell types. So this is, again, where FITZ-Z is the protein that enables cell division, and so you have this protein in rings that enclose the cell, or enclose the cell membrane upon cell division, and so pinches it off. So you would imagine that this is involved in cellularization, but it's actually independent of that protein's expression. So you get these different multicellular or cellularization mechanisms. So this is an example here of where you have the tissue, where you have the peripheral cells and the scutoid cells. The scutoid cells are just smaller. We've talked about this before, where there's more constrained cell morphologies, and they're in this sort of arrangement in the middle where they're compressed, and then the peripheral cells are wider and they're along the edge. And so they're under different types of compression. So you get these different types of cells, and then this is where they're imaging FITZ-Z, and they show in the unicell case where there's a ring of FITZ-Z in the middle, and then in the coenocyte it's kind of diffuse cellularization, it's in the middle, peak cellularization, it's along the boundaries in here, and then the tissue, you see FITZ-Z being expressed along the membranes, I guess. And so this is just kind of showing the difference in how it's expressed. So in the unicell case, it's getting ready for cell division, where the unicell is dividing into 2 cells. The FITZ-Z ring is poised for that. But in these other cases, you don't see that sort of need for conventional cell division. You have something else going on. I guess that's what's going on here. And so you have FITZ-Z1 being expressed across different developmental stages, and that's what you're seeing here. So yeah, this is basically, and then finally, archaeo tissue cellularization is triggered by envelope tension. So what we're basically finding is that and this is put in the context of eukaryotes, so evidence from flies and holozoans suggests that the timing of cellularization correlates with a specific threshold ratio between the number of nuclei per cell volume. This is in a syncytial cell before cellularization, and then that cellularization is triggered. So in flies, you have this process, and you get this these nuclei per volume, and then when you reach a specific number of nuclei per volume, you get the cellularization. And it's like we were talking about with limits to the size of a single cell, you get this sort of thing going on. So by imaging HVO cells expressing amnion green PCNA, we quantified the number of replication sites and fluorescence intensity within replication sites relative to cell area as a proxy for N/C. If N/C is critical for cellularization, DNA replication would increase faster than cell area. However, the signal at replication sites remain constant relative to cell area, indicating that N/C does not influence cellularization. So this is again where they want to know more about cellularization and the nuclei per cell volume, which is N/C. And so this does not influence cellularization in in the paleo Archaebacteria as opposed to what we see in metazoans. Next, we tracked single cell parameters such as size, time, and the amount of added cell volume at cellularization, observing a lower coefficient of variance for cell area compared to area added in time, respectively. So this is where they want to look at different things that contribute to cellularization, and they wanted to see if there was some critical cell size that needs to be reached, so before you get cellularization. So they compressed some of these unicells. This resulted in cellularization around 4.2 times faster, with 3.7 times less added area compared to wild type at a consistent cell size. So they were able to induce this in the unicell using compression and getting a regular result. So that means that there's something going on between this sort of compression and these other mechanisms of cellularization where they're not really sure what's going on. But and then so this is figure 4. Tissue cellularization is triggered by coencytic size through a membrane tension threshold. So there's something going on with membrane tension that triggers this process. Okay, so why don't we stop there? And so I know Susan maybe or Dick wants to say something about this, or anyone's welcome to say something about it.
It would be interesting to look at crack formation with regards to where the divisions occur, but other than that, nothing's coming to mind this morning.
Okay.
Yeah,
Yeah.
Brad, this particular archaea genus comes from the Dead Sea in Israel, and it's actually the first one discovered around the 40s. In the 40s, it produces many polygonal cells, and yet I don't see polygonal cells in these tissues. At least at a glance of them, I haven't done a thorough search.
Yeah.
So the relationship to the polygonal cells would be interesting to know.
Yeah.
Okay.
Yeah.
I guess that's my only comment. I did come across this paper a couple of days ago, so I was aware of it, but I didn't make as much as you did of it. I'm not sure what the role of the S layer is. There's a lot of speculation that it acts like a cell wall, but there's no evidence. I've concluded. Have you ever made a bubble raft? Just bubbles floating on top of water?
Yeah.
Okay, you'll notice that if you stick your finger in it wet, that it's very easy to move it around. So it acts more like a liquid than a solid, even though it looks crystalline. And I suspect that's true of the S layer. The S layer can probably move around. It certainly, I've predicted it has different configurations at the edges and corners of the polygons it makes. And I can actually calculate on assuming that there's a different energy for S layer molecules at the surface, at the edges and corners of polygons. I can calculate the shapes. I'm not sure what's going on with the multicellularity, and I'm not sure there's any natural conditions under which you get the compression that they have artificially generated.
Yeah.
There might be some what do you call it? the term that Gould invented. I'm trying to remember it. The one for pre-adaptations.
Like exaptation?
Exaptations. Is that it? Okay. Yes. So it might be one of these things that's developed for another reason, but then prove useful for multicellularity. There's no evidence in collections of these cells or cultures of them of multicellularity, so the compression seems to be essential and artificial.
Would it have anything to do with bacteria forming films?
No, not at all.
Bacterial films?
Bacteria. See, contrary to the general statement, the bacteria do have rigid cell walls, which can support up to 50 atmospheres of pressure in them. Archaea, especially these archaea, have zero. In other words, there's no osmolarity at all, no osmolarity effect in them at all. So we're very, very different from bacteria that way. Bacteria are sort of little high pressure bombs. Okay. And some of them do indeed are multicellular. In fact, there's even one that's obligatorily multicellular. In other words, it forms a little ball of cells, and it reproduces as the ball of cells, so you never see an individual cell. Okay, so if I've got pictures of this in this book. Okay, if anyone wants to follow it up. How should I say? It's a very interesting result, which I take with a grain of salt.
Yeah, I think that's probably a good way to put it.
Okay.
Yeah. All right.
The tissue-like formation would be stronger than individual cells. Like that would be an interesting thing to look at. How that changes the strength of the cells collectively if they're attached to each other.
Oh, and by the way, on the previous stuff on diffusion, as I said, some of the archaea, such as the ones in this paper, form these very flat cells. And with much smaller volume, but with, how should I say, the surface area per volume is much larger. And it's speculated that this might allow them to capture more molecules or more sunlight or something. Okay, because the membranes may be permeable to small ions and other molecules. Okay, but they're very peculiar cells. They exist in saturated salt water. In other words, much higher than ocean water. They have no osmolarity. They have these peculiar shapes. So there's sorts of things about them. And yes, it's true that people believe that eukaryotes came from a fusion of archaea and bacteria. But whether that's related to this compression phenomenon or not is not obvious. Yeah. So I'd say we're still dealing with an unsolved problem.
Yeah.
Okay, I guess that's it. So.
That's good. Yeah, thanks. Yes, that's good. And of course, yeah. So if you're new to the group, you know, there's a lot of terms that got thrown around that maybe you're not familiar with, but the way I view that is just keep attending the meetings, you know, read the paper if you want, and, you know, eventually you'll kind of get it. It's fun stuff. So,
Yeah, Bradley, there's also one thing I wanted to bring up. I made this analogy between shaped droplets, which between shaped droplets and archaea that are polygonal. They have similar shapes, but the size of the shaped droplets has been shown to be as small as 600 molecules. Okay, which is incredibly small. It's too small, obviously, for much metabolism to occur in, if it developed from that. But it's suggestive that the lower limit for the size of the cell might be much smaller. Okay. In other words, you can get little triangles of these shaped droplets down to 600 molecules. Okay, which is incredible. Okay, so I leave that question open also.
yeah. All right. So one final thing I wanted to mention before we go: if you're doing Google Summer of Code applications, your proposals, those are due, I think, today. You'll have to check the date on the website. I think it's the 7th. And then I don't remember what the time is, the deadline. I think it's UTC. So please check with your local time to make sure you make the deadline. And then we'll be evaluating applications and then making decisions by, I think, May 1st. Oh, April 8th, 1800 UTC. Good. So it's tomorrow, or I guess for everyone's tomorrow. So please get those in on time to be considered, and then we'll be making decisions. I think they're released like May 2nd. I don't know what the date is, but and then, you know, if you don't get selected, because we don't know how many slots we're going to get, then you could still pursue your project, you know, without being formally selected for GSOC. There are ways we've done this in the past with students, and so you know, don't feel too bad if you don't get selected. Sometimes we only get one slot, so it might be you might not get selected even if you have a great application. So good luck to everyone, and look forward to seeing your proposals. And so that's all for today. Thank you for attending. See you next week.